Let's talk for a minute about family and genetics. Imagine you're studying how genetic mutations can occur in family lines. For this class, we'll still keep this simple by examining a type of genetic material that is only inherited from mother to child. I've put the mother, Rosalind, at the top of this family tree. Another name for a family tree is genealogy. Let's say that after having this genealogy, we use genetic technologies to sequence the family member's DNA. Maybe we're sequencing an area of the genome that we consider important. Maybe this area of the genome encodes for an important gene related to breast cancer or some other disease risk. When we compare Rosalind's DNA to her daughters, Gertrude and Marie, we notice that Gertrude's DNA sequence remained the same, while Marie's DNA sequence has changed by a single nucleotide base. A C, signifying cytosine, changed to a T, signifying thymine. In other words, a mutation has occurred. We have placed a pink dot on the genealogy to identify the lineage where this mutation occurred. Now let's imagine we extend out our genealogy to include Rosalind's grandchildren, Jane, Rita, and Margaret, great-grandchildren, Maria, Elizabeth, and Christina, and great-great-grandchildren, Rachel, Barbara, and Carla. Notice that we have recorded 2 additional mutations in Rosalind's descendants. One occurred in her daughter Gertrude's descendants, where the gene carried by Jane had a mutation in which a G, for guanine, became an A for adenine. For this area of the gene, no other mutations occurred in Gertrude's descendants. The other mutation occurred in Marie's descendants. Specifically, we record a mutation in Christina's version of the gene, in which an A for adenine became a G for guanine. This is the second mutation to have occurred in Christina's family line since their ancestor, Rosalind. If we look at Rosalind's great-great-grandchildren, we can count the number of mutations that occurred between these children and their common ancestor. For this area of the gene, there have been 2 mutations since Rachel's and Barbara's common ancestry to Rosalind. However, Barbara and Carla have had only 1 mutation between them. Also, there have been 3 mutations since Rachel's and Carla's common ancestry. We can also see from this example that overall, people who share more recent common ancestor will tend to have fewer genetic differences than people who have a more distant common ancestor. One important concept in genetics is that we can use genetic information to understand ancestral relationships without actually knowing all the exact details about the family tree. Let's imagine a slightly different scenario in which the only information we have is the genetic information from Rachel, Barbara, and Carla. In this scenario, we need to pretend that we have no knowledge of the true genealogy. If we have no knowledge of the actual family tree, we can raise 4 alternative hypotheses for how Rachel, Barbara, and Carla are related. We will label these hypotheses A through D. As a visual aid, I'm going to show these hypotheses as trees. Hypothesis A shows that Barbara and Carla share a common ancestor before either do to Rachel. To explain, notice that in the tree, the branches leading to Barbara and Carla join up. The point at which these branches join is called an internal node. These nodes can be thought of as hypothetical ancestors. So in hypothesis A, Barbara and Carla share a node that is not shared with Rachel. You need to go deeper into the tree to find the node that connects all 3 women together. Given this information, what is hypothesis B proposing? Hypothesis B is proposing that Rachel and Barbara share a common ancestor before either do to Carla. Hypothesis C is proposing that Rachel and Carla share a common ancestor before either do to Barbara. And hypothesis D is tricky. It's proposing that all 3 women are equally related. Now that these alternative hypotheses have been proposed, which provides a better fit to the data? We can see that from our DNA sequencing, there's only been 1 difference between Barbara and Carla. That is, only 1 mutation is observed. All other comparisons have had a larger number of differences. Therefore, the best fit to the data would be a tree in which Barbara and Carla have a common ancestor prior to their common ancestor with Rachel. In other words, from the available data, tree A is the best tree. Once we're comfortable with using trees, we can summarize genetic information quite easily. Here I provide a small amount of additional complexity to the tree. This new complexity takes into account the branching structure known as the topology, but also the branch lengths. The branch lengths here reflect a measure of genetic differences. Even after adding branch lengths, the overall topology still remains the same. That is, we still see that Barbara and Carla share a common ancestor before either do to Rachel. But we also see that sometime after Barbara and Carla's common ancestor, an additional mutation occurred in Carla's lineage, leading to a longer branch length. If you understand these concepts so far, then you can see that trees are very useful tools for showing the nature of genetic relationships. The scientific field of study known as phylogenetics uses these trees to discover and understand much deeper ancestral connections.